Now we're going to talk about vision and how the eye works. Well, the visual axis is the pathway of light. And when you are focusing on something like a clock on the wall, okay, that light is entering your eye at the most direct angle. Um, Things in your peripheral vision, like around on the wall and such, those are entering into your eye at different angles and being projected onto the retina at different places. But what you're focusing on takes the most direct path. Light will enter in, and you should know the structures through which light passes in order to get to the place where we have a receptor for that light. And the unit of light is called a photon. A photon passes first through the cornea and then through the aqueous humor behind the cornea, passes through the opening called the pupil, and then passes through the lens. Then it is passing through the vitreous humor or vitreous body, and then it ends in the back of your eye at an area called the fovea centralis. And there's even a little divot in the back of the eye in this area. Okay, we're going to look more closely at what's going on in that fovea. The fovea has got a dense area of color receptive cells. The color receptive cells are called cones. And not only are they for color vision, but they are for your sharpest vision as well. These receptors require a lot of light to work. Okay? They only work in bright light. So it makes sense that we're going to put them in the path, the most direct path of light, so that they're projected there. Other receptors you have are called rods, and these are named based on their shape. We'll look at that in the next thing, in the next picture. They are very sensitive. They're more sensitive than the cones. They can work in very dim light, but they don't provide color discrimination, and they don't provide as sharp an image, okay, as the cones do. And the rods and cones are going to synapse with other cells called bipolar cells that are going to synapse on ganglion cells that are then going to go on out to the optic nerve. Okay, so there's actually several layers of cells that we need to get to. Notice here, this is a piece, a section out of the back of the eye, okay, near that fovea centralis. And here, notice that light is going to pass first through the choroid, right? Choroid's in there. We had to pass through the vitreous humor. We're going to hit the choroid. And then we're going to come down here and, oh no, that's not choroid, sorry. No, this is a blood vessel in there. Okay, and then we're going to pass through several layers of neural cells before we get to the receptor cells here, the rods and the cones. And you can see the rods are kind of cylindrical shaped and the cones, they taper off, and that's how they got their name. And then there is a pigmented layer of the retina behind them. Okay, so light actually has to go through all these other cells before it even hits the photoreceptor cells at all. Okay. Now, the cones, we have about 6 million of them per eye, and most of them are concentrated there at that fovea centralis. And the area around the fovea centralis is called the macula. And there are no rods in the macula because, remember, this is the most direct path of light. This is going to be the path where light hits most brightly. So we're going to put the receptors there that need that bright light. And rods, they can work in less light, okay? So they can get the indirect light. So this is going to be your area of sharpest vision is here in the fovea centralis. And then this graph is showing that as we move away from the fovea centralis, okay, where the density of cones gets less and less and less, okay, as we get further and further away from the direct path of light, but the rods are going to become more and more dense, okay, as we get away from that direct path of light. And then we have the optic disc where there are no receptors at all, and that is our blind spot. Notice that the optic disc is not in the direct back of your eye. Okay, it is a little medial. Okay, and so light doesn't fall in exactly the same place in both eyes. So you're getting the picture in one eye that is missing where that light is hitting on the optic disc in the other eye. So your brain fills in that gap for you. Nice brain, huh? Rods, we have much more. We have about 125 million. These are not numbers that you need to know. You just need to know their relationship. More rods than cones. Okay, now visual pigments. This is where the magic happens. Okay. Visual pigments are derived from something called rhodopsin, which is visual purple pigment. And this is in the rods. And it has two parts to it. It has an opsin molecule. It's a protein, and it determines which wavelength is absorbed. We have different kinds of, different flavors of opsin. And it's going to tell you, it's going to depend which kind of opsin you have is what light wavelength this particular cell is going to be sensitive to. And then we have retinol, and it's a pigment synthesized from vitamin A. That's why vitamin A is important to your eyes. Okay. Then we have the pigmented epithelium. Okay, well, those visual pigments, those are found inside the rods and cones. The pigmented epithelium is kind of the last stop of the retina, okay, of that neural layer. And then we have choroid behind it. Okay, and this pigmented epithelium back here absorbs photons that are not otherwise absorbed by the sensory receptor cells. Okay, this keeps light from bouncing around inside of your eyes. If we, just like a black t-shirt absorbs sunlight and makes you hot, and a white t-shirt is going to reflect some of that sunlight and keep you cooler, then that's kind of what happens. The dark pigment inside your eyes absorbing that light. But if it was just a light layer of neural tissue, white neural tissue, it would reflect that light and it would bounce around your eyes, and you'd have these weird, like, ghostly visual echoes in your eye. Okay, so color vision, they have some opsin in them, okay, but the same molecule is sensitive to different types. So there are opsin molecules that are sensitive to blue, some that are green and red. Okay, and notice that their percentages were more sensitive to red than we are to blue, and then green is the least. Okay, you don't need to know those percentages, but know those colors. And if we fire all three of those colors at once, if they're equally stimulated, then we see white. That blends all of them together. Okay, there is overlap on the colors that they are sensitive to, but typically we call them the red, green, blue receptors. If you've ever looked at an old television really up close, you'll notice that the light emitters on those televisions were red and green and blue, and it was the different firing and different frequency of the firing of those emitters that allowed you to see the colors on a color television. Okay, color blindness exists, and it's because we have cones that don't function properly. Okay, so they can't distinguish certain colors. Now, complete color blindness is pretty rare. Okay, most people have a problem with one or two of the receptor types. Okay, most common is red-green color blindness, so you don't have any red cones, so you cannot distinguish between red and green. And this is often inherited. It is a sex-linked characteristic. The end of ANP2 will make a good example of this, showing how it's mostly males that are affected, and it's very rare in females. And about 10% of the male population is colorblind, where less than 1% of females are. And you see only 1 in 300,000 people have no pigment receptors at all. All right, now we need to talk about the process of photoreception. I'm going to go through this a little bit in detail here because it's interesting, all right? In the resting state, okay, this is in the dark, okay? In the dark, okay, we have gated sodium ion channels, okay, that are open, and sodium is flowing into the cell with its positively charged ion. Okay? And when we have light absorption and that resting state, notice the potential here, negative 40 millivolts. Well, that seems like threshold, right, for a neuron, right? But this is its resting state. This is the the non-stimulated by light state. That's odd. And then when we absorb light, okay, retinol is going to change its shape. Retinol changes its shape, and here it is in the, you don't need to know this cis and trans state, okay, but just know that it's going to change its shape, okay, and that's what activates this protein. And when opsin is activated, okay, opsin activates a transducin, which then activates this phosphodiesterase, which is an enzyme. And phosphodiesterase is an enzyme that's going to break down cyclic GMP, which is actually a nucleotide. And sodium channels get inactivated. Okay, so cyclic GMP is going to act as our second messenger here, and it's going to shut off the sodium channel. Okay? It's going to put a cork in the end of it, so no more sodium can flood in. And if sodium's not flooding in, we have, we have sodium potassium ion exchange pumps going on, right? Now the changes in sodium concentrations lead to a hyperpolarization, meaning it gets more negative, and decreased neurotransmitter released to adjacent bipolar cells. Look at the potential in this active state. It's now negative 70 millivolts, which is our resting membrane potential for a regular old neuron. Moral of the story, in the dark, your rods and cones are firing, and in the light, okay, they cease firing. So it kind of works backwards, okay, that we are these neuron cells, okay, these receptor cells are more active in the dark than they are in the light. Okay? And this change is then picked up by neurotransmitters, by these bipolar cells, and it's the bipolar cells that are now going to fire on other ganglion cells and take that information to the brain, and that's going to give us vision. And I don't know why. Okay, there we go. Now I can click. Okay, so rhodopsin cannot respond to another photon until its shape is regained. We have to bend it again, and that's called bleaching. Okay, and it requires ATP, and it requires some time to reassemble opsin into rhodopsin. Okay. Okay, so I will see you in the next video, and we will talk about the visual pathway in Skull Session.